Hi everybody, I wanted to go ahead and make a quick video for Chapter 3, Neurocommunication, because it can be a little bit confusing. I'm going to keep my video off because I feel very poorly and I don't want to have to brush my hair and all those kind of things when you don't feel good. So I'm going to go ahead and get started with this video. Something that's going to be important that we'll talk about when it comes to neurocommunication and the membrane of the cell, the neuron cell, is ions. Ions are charged particles, and the most important ones that we're going to be talking about, at least for the first half of our book, are sodium, potassium, chloride, and calcium. And what you'll notice here is that sodium and potassium have a little plus next to them, which means that they are positively charged ions. Chloride and calcium, or chloride is also a charged ion, but it is a negatively charged ion, and calcium is a positively charged ion. So what does that really mean? Well, that means that these ions can influence the overall charge of a cell. And we'll take a look at the details of how that happens. Well, how come some things are positive and some things are negatively charged? Well, that has to do with their basic structure. When we look at the protons and neutrons, which are in the center of the cell, see these guys, these are the protons and neutrons, and we look at the number of protons in relation to the number of electrons. So here's our protons and neutrons in relation to our electrons. That's going to help us determine whether something is a positive or negatively charged ion. So when we look at something like sodium chloride, which is salt, sodium chloride, when it's bound together, doesn't have a charge. So when these separate in our bodies, in our cellular fluid and extracellular fluid, these things come apart. And when they come apart, we see here that as they separate, the sodium ion loses an electron to the chloride ion. So what this means is that the chloride ion now has more negatively charged electrons compared to the protons and neutrons in the center. So it will take on a negative charge. Whereas our sodium has recently lost a negative charge. So now when we look at the protons and neutrons in relation to the electrons, we see that we now have an overall positive charge for this particular ion. We're not going to get into a lot of detail about this. You just need to basically know that some of these things are positively charged and some of these things are negatively charged. Okay, and for the initial stuff that we're going to be talking about, chloride is really the only negative ion that we're going to be talking about. All right. Okay, another thing we're going to be talking about is our cellular membrane and membrane potential. This here is a cellular membrane. It is called a lipid bilayer, is what it's made up of. These are like little lipids, little fatty things, molecules, and basically they form a barrier between the extracellular fluid, the fluid outside the cell, and the intracellular fluid inside the cell. So the job of this membrane is to monitor what goes in and out of the cell. And when this cell is at rest and nothing's happening, we have something called resting potential. And the resting potential is usually a condition where the intracellular fluid, the fluid on the inside of the cell, is more negatively charged than the fluid on the outside of the cell. So if a neuron is at rest, it's not doing anything, it's just hanging around, the outside, the fluid outside surrounding that cell is going to be more positively charged, having more positively charged ions out there than on the inside. The inside overall charge of the neuron would be more negative as it has more negatively charged ions on the inside. So once again, this membrane serves as a barrier to help control the flow of things inside and out of in and out of the cell. At resting potential, usually the inside of the cell is approximately negative 70 millivolts. It differs depending on the type of cell, but for most of the things that we're talking about, we're just going to say negative 70 millivolts is resting membrane potential, for our cell. Because remember, we are all electrical creatures. The cellular communication that occurs within our body happens to be done with chemicals and with electric charge. So that's what this little voltmeter thing is showing you here. Oh, gee, it's at negative 70 millivolts, which is resting potential. All right, let's keep going. Here's another look at our membrane potential, but this time we have some different ions popped in here. So if you take a close look here, these little green ones, we see tons of these little tiny green ions, and these green ions are actually our sodium ions. Try to get my pointer thing here. Oh, there we go. These are sodium ions. And as you can see, there's just a lot of sodium outside the cell, and there will be a little bit inside, but not so much. Kind of think of the cell as like a kind of a tentacly water balloon. That's kind of the way you can think of a neuron, to where it's, has the liquid on the inside. There's also liquid on the outside, but there's differences in the charges and with the different types of ions on the inside and outside. So we also notice here that we have a lot of these negatively charged chloride ions out here in the extracellular fluid as well. However, they're not as much of this chloride as there is the positively charged sodium. We also notice that we have a few of these little yellow ones. These yellow ones are potassium, so we do have some positively charged potassium ions also out there. Now on the inside, what we have are these big protein molecules, and these are just different types of protein molecules that have a negative charge. So these guys are pretty big, and their negative charge is pretty strong. We also see that there's a lot more potassium inside of the cell than there is outside of the cell. A lot more of these little yellow guys on the inside than there are on the outside. And that's a very important thing that we're going to need to remember, is that there's tons of sodium on the outside, kind of wanting to get in. You can see how it's kind of lining up over here. And we have a lot of positively charged potassium on the inside. And those are two very important ions that we're going to be talking about when we talk about cellular communication. You'll also notice that there's a giant blue protein right here. And this giant blue protein that is embedded into this cellular membrane is to allow sodium to flow in and out of the cell. So there are, along this cellular membrane, there is, we can consider the cellular membrane as being selectively permeable. In other words, there are times when it's going to allow certain things to go in and out of the cell. At one time, it might say, okay, we can let lots of sodium into the cell. And at another time, you'll have another protein in here that acts as a sodium-potassium pump that will say, okay, it's time to kick out all of this sodium. And it will pump that sodium back out and maybe draw in some more potassium. So these are little protein structures that are within the cellular membrane that help regulate what ions are passing through the cellular membrane. And this is done in some different ways, and we'll talk about the different ways that this is done. So once again, sodium, there's more on the outside. Chloride, there's more on the outside. Potassium, there's more on the inside of the cell. And there's a lot of large positively charged ions, or proteins, actually, on the inside. I should say protein. All right. I think that's it for this slide. Let's go ahead and move on. As I mentioned, the cellular membrane is really important for deciding what is going to come into the cell, what can go out of the cell. And when the charge of that membrane, of that cellular membrane changes, it changes what it allows to come in and out of the cell. So if the cellular membrane is depolarized through what's called depolarization, the neuron membrane becomes less polarized, in other words, less negative. When we talk about depolarization, we're talking about it becoming less negatively charged. This is caused by sodium ions entering into the neuron and voltage-gated sodium channels are being opened quickly. And we'll talk about the different types of channels that are in the cellular membrane that allow things to travel in and out of the cell. Repolarization is when the neuron membrane becomes polarized again. So in other words, it comes back a little bit more positive to where it was to start with. It goes back to where it was. And this is caused by potassium exiting the neuron through potassium channels that open more slowly. So the movement of sodium and potassium is key to the process of depolarization and repolarization. And I will try to do another slide here to help you understand that a little bit better. Okay, to understand depolarization and repolarization, we have to understand how things can move through that cellular membrane. So I'm using this little drawing pad that I'm hoping to figure out how to use better. Anyway, so let's draw a little bit of the cellular membrane here. There's our lipid bilayer. Here we're going to insert a couple of proteins. This one is going to be an ion channel specific to sodium. And then let's put another one over here. Try not to click the little thing on this drawing pen because it erased everything I did a minute ago. Okay, here's another ion channel. And this channel is going to be specific for potassium to move through. Now, these two channels that I have drawn right here are ion channels. They're a big protein that allows these ions to flow across this cellular membrane. So in this particular case, sodium can move inside or out with this particular channel. Same thing with the potassium. It can move freely back and forth through these channels. So these are what you would call a passive channel. And so these, once again, it doesn't take like energy to move these molecules through here. So what we're looking at here is, why are they moving then? Why does the potassium want to move in? Why does the sodium want to move in? What's going on here? So if you remember from a minute ago, we have a lot of sodium on the outside. All right, we have all these guys floating around out here, a lot of sodium molecules, and on the inside we have a lot of potassium. I'll learn to draw with this thing, but whatever, it should get the point across anyway. A lot of potassium. We also have a bunch of negatively charged protein up along here, and they kind of like to come up towards the membrane, because if you've ever played with magnets, you'll know that these opposite charges attract each other. So one of the reasons why the sodium may want to move into the cell, and why our potassium might want to move out of the cell through these passive channels is, let's see, almost erased the whole thing again. Electrostatic. Pressure. Okay, so that's what we see, like when we have, if you play with magnets ever and you put the two positive sides together, they repel each other. You put the negatives together, they repel each other. Same thing here. These positive sodium ions are going to be attracted to the charge of these big negatively charged proteins on the inside of the cell. So they're naturally going to want to flow into the cell. The potassium wants to flow out, but it wants to flow out for another reason. The reason the potassium wants to flow out is due to diffusion. I'm so sorry, guys. I'm sick. I'm just trying to get something for you here. Diffusion. So when we're talking about diffusion, things want to go from an area of high concentration to an area of low concentration. So in other words, there's a lot of potassium here. So this potassium is going to tend to just want to flow out passively. It wants to diffuse out into an area that has less potassium, and the sodium wants to diffuse in. So this is something that's kind of going along all the time with the cellular membrane. Well, there's another structure here, another protein in here, and this thing acts as a pump. So this thing is what we would call the sodium-potassium pump. Sodium-potassium pump. And what it does is it helps to try to maintain the membrane potential. Remember, we're trying to maintain like a negative 70 millivolts for resting potential, for the cell to just be hanging out at a comfortable place where it's resting. It needs to be negative 70 millivolts. Well, if we keep adding these positive ions in here, the positive sodium ions, it's going to eventually change this polarization and make it more positive than negative 70. And so at a resting state, what the cell is going to try to do is, and during other states, it wants to actively kick out sodium, okay? And it will actually want to actively bring in potassium, because the potassium will help keep the balance, keep it from going too negative. But these sodium ions are going to make it too positive. So that's just to help maintain that balance. So the sodium-potassium pump will kick out three sodiums, 3 out of sodiums, and it will bring 2 potassiums in. And this is a structure that's going to be important later when we talk about action potentials, because this sodium-potassium pump is important for taking the membrane and returning it back to its resting potential after we've had something called an action potential. So I know this drawing is very crude, but it's what we have. So I hope that's helpful, and we are going to move on and talk a little bit more about some of these channels and how they work. Okay, so far we talked about the passive channels, the little passive, let me put a pointer on here, there we go, passive ion channels that work through, that move from one side of the cellular membrane to the other through diffusion or electrostatic pressure. Also, we talked about the sodium-potassium pump, which is going to become important later when we talk about an action potential. It uses ATP, it uses energy in order to move those molecules about. Remember, it kicks out 3 sodiums and brings in 2 potassiums to help maintain the balance and to try to bring that cell back to negative 70 millivolts. Keep it at that particular resting membrane potential. So the next 2 we want to talk about are chemically gated channels and voltage gated channels. So chemically gated channels are channels that are opened due to a specific chemical being present. So for example, let's see, let's go ahead and draw again. All right, so let's put our cellular membrane here, put our protein. Kind of shortcut it a little bit here. Okay, so this particular molecule, this particular channel, we're going to say is a chemically gated channel. Okay, chemically gated channel. So a chemically gated channel is only going to open when the particular chemical is present. So there would be some type of receptor, and say we have a neurotransmitter present that can bind to this. And once the neurotransmitter binds to the receptor, this will open. Okay, this channel will become open at that point. But that is, it requires the chemical to be present. So the chemically gated channels work kind of on a lock and key. We're going to go into detail on this later. We'll talk about in detail the different types of receptors and stuff for this. But for now, we're just going to say that this ion channel is open because a particular chemical was present. So say we have dopamine, which is a neurotransmitter, and say the dopamine is shaped like this, then on top of this receptor, on the receptor, it would have to be able to have the same shape in order to bind. So it could go in there, it would rotate and be able to go in there. For example, if it was serotonin, it would be, say serotonin is shaped like a square, it would have to go into one that had a square. All right, so in other words, if it is a cell, a serotonergic cell that produces serotonin, then the serotonin can be active in that cell. If it's a dopamine cell, then the dopamine can be active there, and we'll talk more about that later. But anyway, you have to have a specific chemical present in order for the chemical gated channel to be open. Okay, so that's chemically gated. And now for our voltage gated channel, this is going to, my computer's going to die. Okay, for a voltage gated channel right here, this is one we're going to talk a lot about in a few minutes, is a voltage gated channel. This one opens when the membrane gets to a certain millivolts. Like say it gets to negative 55 millivolts, then then the channel will open and let sodium rush in. Okay, but it's staying closed until it gets to that particular voltage. So when it reaches this specific voltage, the channel opens, allowing sodium to come in. Okay, that's a good example right there. So the type of ion channels we're looking at are these passive channels. We have our sodium-potassium pump that moves sodium-potassium in and out of the cell. We have these chemically gated channels. That's when a neurotransmitter or hormone or some other chemical is present and it causes a channel to open. And those chemically gated channels only open in the presence of the particular chemical that they are related to. And the voltage gated channels, where they only open up at a specific voltage. So if the membrane is at negative 70 millivolt at resting potential, this is not going to open. So this will not open. The membrane has to get to the voltage of negative 55 millivolts before this channel will open. So that's a voltage gated channel. And we're going to talk more about those in just a minute. Okay, now we come back to depolarization and repolarization. So let's start again with our little cellular membrane here. Get these guys in here. Okay, so we have a channel here. Okay, so this little channel that we have here, this is a voltage gated channel. A voltage gated channel right here. All right, so what will happen is when this cellular membrane here, now remember when we start off, the inside of this membrane is more negative and the outside is more positive. Okay, so that's because we know that the extracellular fluid, because of all of those, we have tons of sodium, sodium floating around out here. Okay, we have a few potassiums, but lots of sodiums. We have a lot of potassium inside. Okay, and we have those big negatively charged proteins over here. Okay, so that's kind of how it's all set up. So at resting potential, this is what it looks like. And inside the cell, we're looking at about negative 70 millivolts. However, if this cellular membrane changes and becomes more negative on the outside and a little more positive on the inside, and we'll talk about how that happens. When that occurs, this voltage gated channel will open and allow sodium to rush into the cell. Now, this influx of sodium into the cell is going to take this part of the membrane here that was negative and cause it to change and become more positive. So now this membrane is more positive, which is going to cause the next voltage gated channel here to open. So now this part, once again, we have sodium rushing into the cell, and as it rushes into the cell, it depolarizes this part of the membrane. Okay, my computer tried to die right there. So let's recap where we are. So at resting potential, our cellular membrane here will be at negative 70 millivolts. So once again, that's negative 70 millivolts. The inside of the cellular membrane will be more negative than the outside. The outside will be more positive. We have tons of sodium hanging out here, a little bit of potassium. We have tons of potassium inside, and we have these negatively charged proteins. So let's say that something occurs to change the voltage of this membrane. This membrane has become depolarized. The depolarizing meaning it's becoming more positive. It is less negative than it previously was. So this membrane gets to a particular voltage and opens this voltage gated channel. So for example, it gets to maybe negative 55 millivolts or whatever the particular, it gets to a particular voltage on this particular part of the membrane. When it gets to a particular voltage, this channel will open, and it is only open when it gets to that particular voltage. When that happens, sodium can rush into the cell. Once the sodium rushes in, it depolarizes this other portion of the cellular membrane, causing it to become more positive, changing the voltage of this section of the membrane. When that happens, it opens this next voltage gated channel, once again allowing sodium to rush in. And then we have the same thing happen again here, where our next voltage gated channel right here, we have our sodium just hanging out. Start off with this negative part of the membrane, negative charge on the membrane. However, all of this sodium depolarizes it, causes it to be more positive than it was, taking it from, say, negative 70 millivolt all the way to negative 55, which is more positive than it previously was. And then it opens this channel. And you can see how this would go, just kind of continue on down the line. As this happens, though, as this part here is depolarizing, the part over here starts to repolarize. As soon as this section is done, it starts to repolarize. So at this point, we'll just pop in our sodium potassium pump here. Say we have one of those guys hanging out here. The inside of the cell is no longer at resting potential. It has changed. So that being said, the sodium potassium pump already says, oh no, let's fix this problem. Let's grab some of these sodiums and kick them out. So the sodium potassium pump is going to kick these guys out, and it's going to, also drag in a couple of sodiums. At the same time, some of those passive channels will be activated as well, where other channels that will allow potassium to come back in the cell and move through the cell are activated. But we won't worry about that right now. Let's just worry about the sodium rushing in, causing the depolarization of a section of the membrane. And then as soon as it moves down the line here, so now this next section over here is getting depolarized. Okay, this voltage gated channel has opened, allowing sodium to rush in. So this sodium is rushing in, it's going to depolarize down the line this way. But as this is depolarizing, this section directly behind it here, let's see, let's get a different color. Okay, so this section back here, that's supposed to be red. Hopefully, no, is it going to be red? We'll see. This section repolarizes. So this depolarizes, and while this is depolarizing, the section before it is repolarizing. So in other words, we have this charge happening here for depolarization, and then our sodium potassium pump is doing its job, and these channels quickly close. So these channels that were opened due to the voltage being a particular voltage will close, and the sodium potassium pump and other channels will come into play in order to bring the cell membrane back to that negative 70 millivolts. So that's kind of how that goes. And I just erased everything, so we'll see how this goes. Oh my gosh. Anyway, wish us luck that this actually didn't erase. Okay, this is going to wrap up this little video talking about cellular membrane and the movement of ions. I misspoke in that last frame. I was looking, I'm kind of sick and not doing great, but I said that the sodium potassium pump would like kick out 3 sodiums, and then I said it was bringing in 2 sodiums. Nope, I meant that it was kicking out 3 sodiums and bringing back in 2 potassiums. That will help it to not go too negative. Anyway, so that's it for this little video here. I'm going to go ahead and wrap this one up, and then I'm going to do another one that talks about the action potential. Okay, so I hope to see you guys, and maybe at office hours, or email me if you still need clarification, and we'll go from there. Okay, thanks guys, take care.